Hi guys, welcome back to Biology Videos at TDA. This is Year 12 and 13 work, looking at 12.6, the specific immune system. It can be quite a tricky topic, so I thought I'd go over this one. I would get a piece of paper and a pen. I'll be asking questions, and at the end there'll be some actual exam questions from OCR, and you can pause the video throughout and then replay bits if needed. We're looking at the action of the B and T lymphocytes in a bit more detail, and what the difference between a primary and secondary response is. Okay, so we're doing some interleaving starters. So our first question will be, which type of bonds form between water molecules due to their polarity? And it's C, hydrogen bonds. What are the simplest carbohydrates called? Monosaccharides, that's one sugar. Which of these is not a property of monosaccharides? So it's B, they can't form peptide bonds, that's in proteins. Glycogen is made up of beta glucose subunits. Is that true or false? That's false. It's alpha subunits. And testing for protein, what environment do you need? And name the solution and the components of that solution. Okay, so you need an alkaline solution. It's the Biret test, and we get the alkaline from the sodium hydroxide, and it's copper sulfate solution. Okay, so on to our kind of main part of this lesson. So a reminder of what you should know beforehand, but this will kind of introduce the beginning of this lesson, which is looking at cell-mediated immunity. We're going to go into a bit of detail now on how we get this MHC complex. So, looking at pathogens when they arrive in our body, and how our neutrophils, in this case, or macrophages, respond to that. So if we start at the top here, number one, the first thing that's going to happen is the neutrophil is going to be attracted to the pathogen, and it's going to attempt to engulf it via this phagocytosis that you learned at GCSE. So this pathogen with its antigens on its cell surface is then going to all enter the neutrophil. So that's then inside the neutrophil. That will then trigger these lysosomes to then, which contain digestive enzymes, to then form a complex, and we call this a phagolysosome. So the lysosomes and then the vesicle containing the containing the pathogen is then going to fuse together. The enzymes are going to break down the pathogen, and that's going to leave these antigens. Now this is going to form this major histocompatibility complex. You can write it as MHC in the exam. And these are then going to be presented on the outside of the neutrophil, and it could be a B cell, and we'll come to this later on. And these are going to be useful for recognising the same antigen again in the future. So it engulfs the pathogen, the lysosome combines with the vesicle, with the pathogen inside of it, calling a phagolysosome. That's going to then produce this MHC, this major histocompatibility complex, which is then going to be presented. Now this is now called an antigen presenting cell, or APC, because it's presenting the antigen from the pathogen on the cell, these little red triangles. Okay, so that's kind of the last lesson. Looking at this lesson in a bit more detail then, what are antibodies? So you need to know what antibodies do, and you need to know the structure of them. So we'll start off with what they do. So first of all, they act as opsonins. So these are the antibodies that will tag onto pathogens, and that will then cause the phagocytes to engulf them. We've then got the antibodies will just simply bind, making an antigen-antibody complex. They'll bind to a pathogen, which will then often make them not dangerous. It will make them stop working. They can act as agglutinins, so they're going to then cause the pathogens to clump together, and that's going to make it easier again to break it down by phagocytosis. And they also produce these antitoxins. They're going to bind to toxic molecules and then prevent them from being dangerous. So this is what we have. We have a glycoprotein. So antibodies are all the same shape, however they have a different variable region. So I'll start off with kind of the main thing bit. So this kind of dark pink here, you've got these heavy chains, and they're going to be held together here. These little black lines are representing disulfide bridges, so you need to know that. So disulfide bridges are going to keep the two heavy chains together, and then you can have these variable regions at the ends of the antibodies. Now there are about 110 amino acids between the light, which is the outside one, and the heavy one, and they're going to be specific, a bit like the lock and key model in enzymes. They're only going to fit the right shape of the particular antigen which binds to it and makes that complex. Okay, so you need to know the overall shape of it, you need to know it's a glycoprotein, you need to know it has a variable region at the top, and they have these disulfide bridges. Okay, so a few terms, we're going to watch an animation shortly. A few terms, and you can pause the video, make notes if needed. So an opsonin, a reminder they bind pathogens and tag them, that's the antibodies. We're going to have these cell signalling molecules called cytokines, particularly one called interleukin 1 and interleukin 2, we'll look at those later. Antibodies, you should now know they're Y-shaped glycoproteins, and then agglutinins, they cause pathogens to come together. Okay, so I've done my best here in the next two slides to summarise all the different types of T and B cells. I would pause the video at some point, make notes on this, and then on purpose try to learn them, perhaps try and write it off by heart, say it out loud, get someone in the house to quiz you, etc. So we have, let's start off with T cells. Now T cells are kind of the ones that go ahead and do the killing of the pathogen quite often. They're going to be made in the thymus, it's quite easy to remember. The T cells are made in the thymus glands, and they're going to carry out the cell-mediated immune response, and they're going to be recognising the abnormal cells and killing them. They're often involved with things like viruses and cancers. The B cells are made in the bone marrow, again nice and easy to remember, B for bone, B cells. They're going to mature there, they're going to carry out the humoral immune response, and they're going to be producing the antibodies, so this is going to be outside of cells. So, what does each one do? So we've got the plasma B cells, and we'll start there. Now these are the ones that are going to produce the antibodies. They produce up to 200 per second, which is really, really lots and lots. They're only live for a couple of days, and they're going to do this really, really fast secondary response, which we'll get to later. You've got these B effector cells. They're going to then divide to form these B plasma cells, so we need to have a stock of these to make sure we're producing lots of plasma cells. And then the memory cells, because the B plasma cells don't live for very long, they live for a few days, but they produce lots and lots and lots of antibodies in that time. We're going to need to remember these pathogens when they get to us later on again. So they're going to, excuse me, remember the specific antigens. Right, so the T cells now, we're going to look at the helper T cell in a bit of detail. They produce interleukins, which are cell signaling molecules which are going to get other cells to do various different things. The killer T cells, often referred to as natural killer cells, they're going to produce these, or cytotoxic cells, they're going to produce this protein called perforin. Now, this is kind of the killing of some of the pathogens. They're going to make holes in the cell membrane of the pathogens, and that's going to cause a massive exchange of substances inside. It's going to ruin the cell, it's going to kill it off. These memory T cells, so they're a bit like we've got the memory B cells, they're going to then be a stock, and they're going to divide rapidly to make the killer T cells. And then the T regulator cells, they're going to be used to suppress the immune system, because we don't want an autoimmune response on healthy cells. So we're going to start off by watching an animation. There's going to be some questions afterwards, kind of going over what I've said, and then I'll talk over the animation in my own version. So I'll just move my screen capture.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin 1, which stimulates the T helper cell to secrete interleukin 2. Interleukin 2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Okay, feel free to replay that if you need to. So here's some questions. I'll let you pause the video now, have a go at these. Okay, so what type of cells ingest pathogens? So macrophages, I'd allow neutrophils as well. What type of cell interacts with antigen-presenting cells? So the T helper cell, we'll look at those more in a minute. What does a cytotoxic T cell do to an infected cell? So that's the killer T cells we were talking about earlier. So they bind to them, they produce chemicals, so they're perforin. B cells are activated what type of cells? So T helper cells. This causes them to differentiate into what type of cells. So we've got plasma cells. What type of cell produces antibodies? We've got plasma cells. What type of cell helps provide immunity? Memory cells. Okay, so I'm going to try and break this down, this information, a little bit more and a little bit slower. So a reminder of some key terms here. So cell-mediated immune response. So that is breaking down things which, killing pathogens which enter cells and destroy cells from the inside. So we've got examples of the virus or cancerous cells or transplanted cells, and they recognise the antigens on the cell surface of these. A reminder that these T cells mature in the thymus gland, and you can see they kind of situate just below the lungs. And we also know that it will shrink if a person is under a lot of stress. So if a person is particularly stressed, their immune system can be down, it can be weaker, and this is partly because the thymus gland will shrink. How do they kill them? They produce this protein, so the killer T cells, they produce this perforin, which breaks down the cell surface membrane, and that's going to allow the movement of substances in and out, which is going to then kill the cell. Okay, so a reminder of that then, kind of an image. This is taken from the Caboodle textbook. We've got, which we looked at earlier, we've got our MHC, which is then going to form and produce an antigen-presenting cell. So the T helper cell is going to be started by binding to the receptor of this antigen-presenting cell, and then you're going to have the interleukin-1 and interleukin-2 then released. So interleukin-1 produced from whichever cell this is, and then that's going to then get the T helper cell to then produce interleukin-2, and that will then become a mature T cell. And if we're going to clone this one, it's going to be then by mitosis. This can then do lots of other things, and we'll come to this in a moment. So that could develop into more memory cells, produce interleukins again to get other cells to do things. This could cause the B cells to divide, and then this could develop directly into T killer cells, which can then produce things like perforin and kill them off. Now let's have a look at the humoral response. So that was cell mediated. The humoral is making the antibodies, making the B in the plasma cells. And in the body we have a lymphatic system. So most of the time our plasma cells will live in the lymphatic system and not in the bloodstream. And these little dots are lymph nodes. You can sometimes feel the ones in your neck if you're unwell, or under the armpit. And we've got the thymus and the spleen, which is also part of it as well. Okay, so I'll do my best to summarise then the humoral response. So whether it is coming from an antigen-presenting cell or whether it's from an activated T helper cell, the B cell is going to become activated. And when this B cell becomes activated, we call this clonal selection. So we're selecting the B cell that we want to use. That's a really important word to remember. Then what's going to happen? We've selected the cell which is going to produce the right antibody, or we're going to have to make lots of clones of this cell for whatever reason. Then we're going to have something called clonal expansion. So this is when this B cell clones itself to produce either a plasma cell or a memory cell. And this is an example of mitosis, which is quite a common question, which will likely be, apart from growth and repair, give an example of why we do mitosis. So this is an example. We do mitosis in clonal expansion to produce these plasma or memory cells. Now, let's say we've selected the plasma cells. This cell then is going to then divide, and it's going to turn into a plasma cell. It's then going to produce lots and lots of antibodies, which is the primary response, and this is going to break down the pathogen and then allow us to then have these memory cells. Or we could have selected a memory cell, and this could simply be circulating around. We're dividing this to keep hold of memory cells for future infections, which brings us to our primary and secondary response. So, task for you now is, can you please summarise the process of the humoral immunity of B cells? Pause the video and have a go at that. Maybe 6 bullet points. Okay, so this is taken from a Caboodle task, which I've done and selected for you. So one of the ways we could summarise it is the antigens of an invading pathogen are absorbed by the B cell. The B cell processes and presents the antigen on its surface. Right, or the helper T cell is going to attach to the antigen, which activates the B cell. We call that clonal selection. It's then going to divide by mitosis, become a plasma cell. They're going to secrete lots of antibodies specific to the pathogen. Something which I haven't mentioned very much at the minute is you need to make it very clear that when we're having these antibodies, they are specific, and we use the word complementary to say they're complementary to the pathogen or the antigen. The antibodies are then going to attach to the antigens and destroy it, and then some of the B cells are going to develop into memory cells to remember for future infections. So this is where you hear the sort of common phrase of we remember when it comes, when a pathogen attacks us, we remember it for the future, which is kind of what the current government is trying to do with the coronavirus, is they're trying to hope that we're going to have this immunity by producing these memory B cells. Okay, so the primary immune response. This is what we've just looked at. So this is the first time we're exposed, and you often see this in a graph. First of all, it's often a lot slower, and you have to know the difference between why the first one is slower and the second one is much more rapid. So there's a delay because we have to produce these antigen presenting cells. We have to do the initial working out of that it's a pathogen, and we have to present it. So this takes a little bit longer, and then forming these memory B cells. Okay, whereas the secondary response, so the second sort of peak, this is much quicker because we already have our memory B cells. They're going to then divide rapidly, so clonal expansion, and they're going to produce many, many antibodies really, really quickly. So the reason why it's quicker the second time is because we don't have to produce these antigen presenting cells the first time. And just a little comparison of the two. You can pause the video and have a look. Okay. So now we'll look at some actual exam questions. Again, keep pausing the video throughout this. So the first one is figure 25. It shows the graph we just looked at. This is a patient's bloodstream following a vaccination. Really important that we extract the information from here. This is following a vaccination. Okay, explain why the response to the subsequent infection is much bigger than the vaccination. So why is this second response much bigger than the initial vaccination? Okay, have a go at that. Okay, so the idea is the vaccination is going to take longer because it is going to involve this selection and then this expansion. It's going to take longer. The memory cells are already present in response to the infection, which is sort of the converse of that. Is the second time the infection comes, it already has memory B cells present. Okay. Question 2. So we've got our Y-shaped glycoprotein. I'll let you have a read. Okay, so just sketch one perhaps. Really simple, we just got to do some lines. Right, draw a ring around, which is the part which is complementary to the shape of the antigen. And then question 2. State what type of bond is found here and give it its function. So what type of bonds do we have in these glycoproteins? And what is the function of them? Okay, pause the video. And the answer, so you should have circled, for the first section you should have circled around the top. This is the kind of things you were allowed, so you could have just circled one side, although it should be both. This is the best answer you could have put, or maybe 2 circles around each side. And then for question 2, what type of bond is found there? We've got the disulfide bridges, which is going to hold the heavy and the light chain together. And the final question then, a little bit more difficult, outline the processes that lead to the production of antibodies against an unfamiliar bacterium. And 2, explain how T helper cells act to speed up these processes. So the T helper cells we looked at earlier. Pause the video and have a go at these. Okay, so for the first question, outline the processes. So the kind of key concept you're trying to get across here is that there are these specific antigen receptors on the B cells which are going to carry. So these antigen presenting cells, they're going to be on the cell surface. They're going to be complementary to only one antigen. So 1 mark for making that really clear. That's going to be selected, clonal selection. That's going to proliferate. So the proliferation, that word means to divide by mitosis of B cells. It's then going to form plasma effector cells, and that's going to then secrete the antibodies. Question 2, explain how the T helper cells act to speed up these processes. So they're going to be stimulating the B cells. They're going to release these interleukin, specifically interleukin 2, because the cells themselves release interleukin 1. They're going to cause the B cells to proliferate by mitosis, which we call then clonal expansion. So this is really important that we remember clonal selection and clonal expansion. Okay, now you've had a go at that. Can you email me how many marks you've got on the exam questions? It was out of 11 marks. Okay, so can you email me your response? And I'm now going to put this on Show My Homework. So can you put submitted once you've watched this, you've done the activities, and then email. Thank you.